In chapter 1 of the cell, we're going to reintroduce some concepts we had learned about in Principles of Biology 1. In particular, we'll talk about the origin and the evolution of the cells. We're going to talk about how the first kind of cells were originated, how we thought things had come together, different models and such. We're going to talk about how the cell evolved and also how the metabolic reactions had occurred within the cell as well. Some of the concepts, again, should seem very familiar to you from Principles of Biology. Some may take it a step or two further. We'll build upon some information that you already have from previous classes. We'll talk about both the prokaryotic and eukaryotic cells, the major differences between the two, and also with some of these similarities as well. And we'll talk about the evolution of the eukaryotic cells in particular as go from the single cell to the multicellular organism. Now, before we talk about how cells evolved, we first have to talk about the study of organic chemistry, or basically the study of organic molecules that we find contained in the numerous cell structures that we see today. So basically, this is the Urey-Miller experiment back from 1953. Should be very familiar to you. Very similar to what Dr. Miller and Dr. Urey had tried to demonstrate was the primitive Earth. The gases that are found in the atmosphere with electrical discharge from lightning could have possibly arose different types of organic molecules, like different types of amino acids, as well as other metabolic byproducts like urea. So very simply, what they did was they took different compounds of gases like ammonia, methane, water, and hydrogen, and basically did some electric discharge and allowed the areas to cool. And then basically the byproducts that received were the organic molecules you see at the bottom of the slide here, including some very familiar amino acids like alanine, aspartic acid, glucamic acid, and glycine, different types of metabolic byproducts, including urea and lactic acid. So again, this kind of gives a good idea of how possibly these cells may have first arisen. Now, this was actually refined a little bit later, I believe in the 80s, where they had a better idea of what the atmosphere was like back then, but they still came down with the same products out of the entire experiment of the organic molecules you see here. Of course, it might be a little more diverse than what the original Urey-Miller experiment originally had. So now we have an idea of how the possible building blocks were formed to the organic molecules. Now let's look at different macromolecules. So if you look at certain macromolecules, we're looking at one particular type called RNA, which should be very recognizable for you. We're looking at 4 nucleic acid bases that are complementary to each other, cytosine and guanine, and adenine and uracil, that you'll piece together just like the pieces of a puzzle, to form long chains of polypeptides. Now, the very interesting thing about RNA is, if you remember anything from Principles of Biology as well, you know what's known as ribozymes. RNA has the ability to self-replicate. So with this ability, you can build quite a bit amount of RNA that will basically perpetuate it and reproduce over and over and over again, which basically gives a building block or some kind of inheritance from one cell to the next. And as we see a different macromolecule with phospholipids, we see these become encased and basically be protected from the outside environment, so it actually can replicate even more so. Now, the RNA functions like a gene in this aspect where it's going to basically pass down its genetic information down to the next generation of RNA in this case. So if you remember, through both transcription and translation, the RNA can self-replicate and basically pass its original information down to the next generation again. Now, another macromolecule that you're very familiar with is the phospholipid. So the phospholipid is known to be amphipathic, which means it has both a hydrophilic and hydrophobic ends. And what scientists have noticed is when phospholipids are placed in water, they will automatically generate themselves into a bilipid membrane and start to separate itself, just like oil droplets in water, to form these little membranes. So what we perceived that happened was RNA became encased in the phospholipid membranes, which protected it from the outside environment. If you remember from way back in biology, we talked about how subtle the change could be for RNA or DNA or any macromolecule when it's either heated or cooled or there's changed different environments like the pH. Sometimes these particular macromolecules can be broken down very easily. So this phospholipid membrane very simply protects this particular batch of RNA from the outside environment from any kind of degradation. So we established the basic structure for the cell: phospholipid bilayer membrane, as well as some kind of genetic material, in this case RNA. Now we have to look at how the cell could possibly perpetuate itself or sustain itself to be able to pass on its genetic information to the next generation. So if you remember from Principles of Biology 1, we talked about different types of metabolic energy processes, including glycolysis, which breaks down glucose into 2-pyruvate and generates 2-ATP. And if you remember, we actually have to invest 2-ATP in order to get 4-ATP, so we net 2-ATP as far as this metabolic reaction. So adenosine triphosphate is going to be the main, not the only, but the main type of energy molecule that we're going to utilize in most of the cells. Very simply because of the bond found between the second and third phosphate. That breakage of that particular bond releases energy that can be used for a different type of reaction. So in this case, glycolysis was probably the first that was that evolved. Even though it's not the best energy resource, it was the only resource that was available at that particular time. Then at some point, we believe that some type of cells were able to provide photosynthesis. It may not have been in water at first. It might have been hydrogen sulfide. But hydrogen sulfide mixed in with carbon dioxide and sunlight can convert carbon molecules into glucose and also therefore give off either oxygen or basically in the very early primitive Earth could have been sulfides, could have been releases, or sulfur in particular. But a more advantageous type of energy generation is through oxidative metabolism, where whether it be either photosynthetic cells or other types of bacteria that are non-photosynthetic, they can basically oxidize the glucose using the presence of oxygen. They can generate carbon dioxide as a byproduct and, of course, water, but generates a whopping 36 to 3 ATP. So that's not an exact amount. Those of you that might remember from Principles of Biology, that also involves the proton pump found in mitochondria that creates a gradient to basically shuttle hydrogen atoms from one area to the next area of the mitochondria, and that shuttle helps generate the pump to produce ATP. So not every hydrogen that is going to be produced from the glucose molecules will necessarily be used for ATP for other reactions. It might be actually used for other reactions all throughout the cell itself. Now we have two domains of cells. We have prokaryotes and eukaryotes. And this should not be new information for you. This should be something very familiar to you. Very simply, the prokaryotes tend to be very simple type of cells where there is no defined nucleus or any kind of membrane-bound organelles. However, there is some nuclear substance found in the cell, known as a nucleoid, where it's a little more condensed in certain areas of the particular cytoplasm. The diameter tends to be a lot smaller than your typical eukaryotic cell. As mentioned before, there's no cytoplasmic organelles, no membrane-bound organelles except mitochondria. DNA content tends to be a lot smaller than what you might find in a eukaryotic cell, but not always. And the chromosomes are going to be bound in a single circular DNA molecule, whereas the eukaryotic cells very simply has a defined nucleus, has a nuclear envelope that separates the nucleus from the rest of the cytoplasm. It tends to be fairly larger than most of your typical prokaryotic cells. It does have membrane-bound organelles as well. There will be partition for different types of purposes, whether it be the Golgi apparatus, whether it be the endoplasmic reticulum, vacuoles, and so on and so forth. The DNA does tend to be quite large compared to the prokaryotic cells, and also going to be bound into linear capsules of DNA molecules, as we see later. They'll be bound in different types of histones. They'll have to be unwound in order to be duplicated and replicated or transcribed into RNA molecules. This is your typical prokaryotic cell, just has some very basic structures. You see a nucleoid found in the center of the cell here, which is going to be condensed DNA material. But the rest of the DNA material will be interspersed all throughout the cytoplasm through here, but it's going to be more condensed in that particular nucleoid. But again, it's very loose. There's no real membrane there to separate the nucleoid from the rest of the cell. You have a plasma membrane, as well as the cell wall that we'll see there as well, which makes the prokaryotic cells relatively hardy against the outside environment. Most of the reactions that we normally talk about in mitochondria or found on ribosomes or found along different types of organelles can mostly be found either found in the cytoplasm itself or in the plasma membrane. Here we see again the two basic types of eukaryotic cells, which should be very familiar to you again. So we're looking on the left an animal cell and on the right on the plant cell. So you'll see a lot of analogous structures here. You see nuclei as well as ribosomes and endoplasmic reticula found on both types of cells. You have a definitive cell membrane found on both cells. However, some key differences found on the plant cell, which includes the cell wall, as well as the vacuoles through here, and of course the chloroplast, which we don't find in animal cells. Here we see a representation of your typical animal cell. Rather simplistic, there are a couple things that are missing from this particular animal cell. In particular, we talk about the cytoskeleton in different parts. If you remember from Biology 1, there's different types of size of filaments, including microtubules, intermediate filaments, and so on and so forth, and microfilaments that make up the cytoskeleton. They're kind of missing with any kind of detail here, as well as cilia, if present, as well as flagellum or flagella if there's more than one, found in sperm cells found in humans, as well as bacterial cells with multiple flagellum. So very simply, we see the nucleus, which is well defined with nuclear material of DNA. It's going to have a continuous membrane with the endoplasmic reticulum, which you'll see both rough and smooth. Rough, of course, has ribosomes attached to the surface, whereas the smooth does not. And we find ribosomes both in the cytoplasm itself, free floating, as well as attached to the endoplasmic reticulum. You find the nucleolus, which is the more condensed packed version of the DNA material. We see a mitochondria in there. Of course, usually we see multiple mitochondria in most cells. You'll see the Golgi apparatus, which is the major area to package, sort, and also export different types of products, to be put either into the cell, the plasma membrane, or exported out of the cell, or simply processed through different parts of the cytoplasm. Here we see your typical plant cell, which will have a couple extra structures here that we do not normally see with animal cells, including the chloroplast, which we know is very important for photosynthesis, the vacuole, which is going to be a major site for either water storage, different types of molecular storage, or different types of enzymatic reactions. You also see a definitive cell wall, where the plasma membrane will be abutted right up against to it, as long as there's sufficient turgidity or enough fluid found within the cell. Use the plasma membrane is going to be right up against the cell wall. If you remember from way back when, basically if there is loss of water, then very simply the plasma membrane starts to pull itself away from the cell wall and starts to degrade the actual cell itself. But we find the same things here that we see in the animal cell, including the nucleus and the well-defined nucleolus. You find the membrane is going to separate it from the rest of the cytoplasm. Also continuous with the endoplasmic reticulum. You only see one mitochondria here, but of course there's usually multiple. You have the Golgi apparatus that has the same type of structure as before. And one other thing I'll mention too is deep within each of the cell walls, you have what's known as plasmodesmata, which are little gap junctions that allow different types of gases, products, or even waste products to be passed back and forth from one adjacent cell to the next. And this could be gases such as carbon dioxide or oxygen, or different small micromolecules that can be passed through. Oftentimes you'll see different types of veins that will pass very close to the plasmodesmata to allow transport of different types of substances. This slide gives you a good representation of how the cells evolved from the initial first cell. So we see three domains here. We have two prokaryotic domains in the bacteria and archaea. Main difference between the bacteria and archaea, both being prokaryotic cells, the archaea are found in more extreme environments, whether it be in undersea volcanoes or different types of vents, usually in very high and or low different types of temperatures. And at some point, the archaic type of cells had other plasma membranes that were basically invaginated, found within the eukaryotic cell. So now we have a membrane-bound organelle, and tough to say which one was first, whether it be nucleus or whether it be the Golgi apparatus or so on and so forth. And at some point with the eukaryotic cells, there were different types of bacterial cells that basically infused themselves as mitochondria or chloroplasts into different types of eukarya. And that's how we started to have different types of eukaryotic cells will develop into multicellular organisms, which will oftentimes sometimes be unicellular at first and then evolve into multicellular. And some we find now, which includes different types of amoebas, will oftentimes, if the situation is very advantageous to them, they'll stay as unicellular organisms. But if food is scarce, they'll actually combine the cells into a multicellular organism, basically for survival purposes. But very simply, the eukaryotic cell at some point will have some kind of invagination of a second plasma membrane, which will help separate the individual organelle to the rest of the cytoplasm. And also, the animal cells will invaginate cells with mitochondria. And then further on down the line, we have plant cells that invaginate cells with chloroplasts on top of the mitochondria, which we'll talk about in the next slide, which is very unique with the mitochondria and chloroplasts. We see different types of DNA found in there versus the actual DNA found within an individual eukaryotic cell. Here we see an example of endosymbiosis, where an archaea cell is going to consume a bacterial cell, but the bacterial cell will remain intact. So the bacterial cell's own DNA will, for the most part, be integrated into the host genome with a small subsection there left to itself. So over time, these bacterial cells will turn into mitochondria. And of course, as you know, the mitochondria is going to form a symbiotic relationship here with your archaea cell by taking over the oxidative metabolism to generate a multitude of ATP for the host archaea cell to use. So if you remember from biology, we talked about different types of metabolic processes. Glycolysis is done in the cytoplasm, where oxidative metabolism is found in the mitochondria. So you can see very simply that that metabolic byproduct of the glycolysis found in the cytoplasm is actually it's going to come from the archaea cells, whereas the bacterial cell gives the benefit of oxidative metabolism in and of itself. And another point to make too is when you look at different types of informational processes found in the cell, especially eukaryotic cells like DNA replication, transcription, protein synthesis, this was all derived from archaebacteria. Whereas most of your basic cell operational processes, which includes glycolysis, amino acid biosynthesis, oxidative metabolism, is all going to be derived from bacteria. Here we see a very basic eukaryotic cell known as Saccharomyces cerevisiae, otherwise known as yeast. So each individual cell that you see here is its own self-contained organism. So it is unicellular, even though it is eukaryotic, because it has its own membrane-bound cells. Now, the actual genome is only slightly larger than most bacteria that you find. So even though it's a eukaryotic cell, the genome itself is not much larger than you find in some of the larger prokaryotic cells. We take our next step up into complexity into the unicellular eukaryotic cells, with the paramecium to the left and the chlamydomonas to the right. The yeast that we saw in the previous slide is not a movable cell. However, those of you might remember from biology, the paramecium to the left is going to have cilia all around the cell wall, which we'll look at a little more closely in a second, whereas the chlamydomonas is going to have two different flagella found for propulsion. Here we can see a little bit better of a picture of an actual individual cell with most of the membrane-bound organelles. You can find the nucleus, which is going to be found off to the left, the white center there, whereas on the opposite side, the darker portion there is your vacuole found in the paramecium, which helps pump water out of the cell, so therefore it won't lyse when it becomes too consumed with water. You see cilia, which is the hair-like projections all around the cell wall and the plasma membrane, which allows for movement found within the fluid. Now, one thing that we have to talk about too is the paramecium. The size of the genome does not necessarily mean very much as far as how complex that particular eukaryotic cell could be. In this case, the paramecium, even though the genome might be smaller than, say, human, the actual coded portions of the actual genome is many times more than we find in humans. So in other words, we have a lot of junk DNA that is not really used in our particular genome, where the paramecium has quite a bit more of, let's say, useful DNA found within its own genome versus our own. Now, unlike the paramecium found in the slide previous, which is going to be considered an animal cell, this is a chlamydomonas. These are going to have photosynthetic cells as well, so we'll have chloroplasts found within. So this is one of the unicellular eukaryotic cells that will have both a photosynthetic component, meaning the chloroplast, as well as typical cells or typical components we find in the animal cell. Again, the genome is quite large compared to most bacterial cells and has more coded portions of the genome than humans do. So here we see the composition of different cell genomes of different types of organisms. Now, I will say this right off the bat, I will not test you on the actual numbers here of which type of organism has the base pairs versus protein-coded genes. You will not be asked for specific numbers. However, I do expect you to know that certain types of organisms, even though might have a very low DNA content of 1 million base pairs, they may have a multitude of protein-coding genes. You can see the general idea, though, if you look at archaeobacteria and bacteria, since they are smaller because they're prokaryotic, versus the unicellular eukaryotes, you see quite a bit of magnitude difference from one to the next. The other thing I want to mention, too, is if we look down under unicellular eukaryotes, the paramecium tetralia, always tough to say that. Basically, the haploid DNA content in 1 million base pairs is at 72 million base pairs, but it has close to 40,000 protein-coded genes versus the next one down. The chlamydomonas is going to have 111 million base pairs, however, only coded for 18,000 protein-coded genes. And those of you who may remember, as far as the haploid versus diploid, the haploid is half of the amount of chromosomes that we have in a particular cell. So we have, let's say, 46 chromosomes. That will be a diploid cell. A haploid cell will have half of that, as 23. To go up in complexity of eukaryotic cells, if you look at the haploid DNA content of Zea otherwise known as corn, has 2.2 billion base pairs and codes for 34,500 genes. Go further down into the animal section, you look at the Downy Auraria, the zebrafish, is 1.4 billion, but codes for 30,000 genes. Mus musculus, the mouse, 2.7 billion, and codes for 26,500 genes. And our haploid content is the highest at 3.1 billion, but only codes for 20,000 protein-coded genes. So you can see very simply that one does not really hold up to the other. So even though you might have a very large DNA content, there only may be a certain portion that will actually be coded for different types of genes to code for proteins. So here's an example of how a unicellular eukaryotic cell became a multicellular eukaryotic cell. This is an example of Volvox, which is a multicellular green algae. So you're going to have smaller somatic cells that you'll see here embedded in this gelatinous matrix. Doing surrounding 16 types of germ cells. So basically, these evolved together, working in a symbiotic relationship based on the availability of food. If food is very scarce, these particular types of cells will embed themselves in the gelatinous matrix to basically protect each on itself and share resources. Now, in some instances, a eukaryotic cell, when it's unicellular, doesn't necessarily always go in the same direction as a unicellular into a multicellular. A lot of times they can go back and forth. So here we see what's known as Dictyostelium discoideum, which is amoeba. So an amoeba, when it's by itself, if the food is plentiful, then they stay as unicellular and they move about on their own, basically trying to pick up food. When food is scarce, they'll actually bound together in what's known as a fruiting body to create spores to protect itself and actually sustain itself under less than advantageous conditions. This is just an enlarged picture of the two unicellular amoebae. So very simply, you see two distinct plasma membranes on either side here. So again, both of the amoebae are more or less independent of each other. So as we look at the fruiting body here, which is going to be comprised of a multitude of different individual amoebae, very simply what we see is the sharing of different types of responsibilities and resources. So very simply, you see certain types of cells will become more specialized. Let's say some on the outer area will be more for protection, whereas more of the cells on the inner surface will be more for, let's say, metabolic processes or for the excretion of wastes. You're basically seeing a division of labor amongst all different types of cells. So this gives rise to what we would known as tissues in different types of organisms, which when I go into nanophysiology, when we look at the different types of levels of organization, we talk about the chemical level up to cellular level, then up to tissue level, which under tissue, we tend to have the same types of cells that basically form a stronger, more uniform type of tissue, which when you add in different types of tissues, they all form different types of organs that we see in different types of organisms. So in this case, we see the fruiting body here. You can imagine your skin is made up of epithelial cells, which you have certain types of epithelial cells which are there for protection against foreign organisms, and some actually are destined to die and give us more of a waterproof outer layer to protect us from the outside environment. So you can see when a unicellular eukaryotic cell will actually form itself with multiple other ones, you'll see division of labor as well as different types of specialization. So some cells become very good at certain types of processes where others become more specialized and actually not quite forget to do other things, but they don't quite do the other types of metabolic processes as well. So here's an example of multitudes of eukaryotic cells working in conjunction with one another in different types of tissues that we find in plants. So very simply, we see three different types of tissue in plants. We see ground tissue, we see dermal tissue, and we see the vascular tissue. So very simply, letter A, the chyma cells, these help provide support. So this will provide the ground tissue. So it gives you thick cell walls, it gives you structural support to hold the plant in place. Under the epidermal cells, this is more of dermal tissue. So this basically gives you a protective coat, but allows enough of absorption through materials back and forth within the cells And finally, have the xylem vessels on the right side here. Will give you the vascular system to transport different types of products, elimination of wastes. But all three different types of tissues here are going to be comprised of the same types of cells found within the tissue, all working for a common goal and a common purpose. Taking a closer look at the Kalenchima cells, so you can see very simply here that they're in somewhat of a rigid formation, and you'll see in between the individual cells outside the actual cellular walls, you have a very stiff, what we call matrix or intercellular matrix, which if you look at different types of ground tissues like roots or trunks, these are built for support and built for resistance to different types of abnormalities found in the environment. So high winds, heat, cold, basically trying to keep all the tissues protected from the outside environment. So here we see epidermal cells, which will allow the passage of gases like oxygen and carbon dioxide, and also allow some sunlight to pass through. You find these mostly on the leaves and the petals of different types of plants, not as rigid as what you find in ground tissue, but rigid enough to basically hold the particular cell constituents in place, but still allow certain things to pass through. And finally, you find the vascular tissue, the xylem vessel elements that we see here. So basically, you find these almost like the veins that we find in different types of plants, allows transports of water, transport of different types of products, as well as wastes, both to the all portions of the particular part of the plant itself. Here's the examples of cells found in animal tissues. So of the four tissues that we have, which includes epithelial tissue, connective tissue, you have muscle tissue, and also nervous tissue. We only see epithelial tissue and connective tissue on this particular slide. On the left, you see epithelial cells. So these will be the lining of the skin or your intestinal tract. You have special epithelial cells here. You see the darkened pink and purple. You see this up against what's known as the basement membrane. So this particular type of cell is more so for protection and abrasion. So what happens is these particular cells, as they mitocondrially divide, they grow towards the surface. You see the flattened area there, and they start to die off. If you notice, there's less nuclei found through there. So you notice that these particular cells form a nice protective layer on this particular surface of the epithelial tissue. So again, you find this usually on the surface of the skin, inside the mouth, and along the gastrointestinal tract. They're constantly mitocondrially dividing, and they are matched up against each other. So you see very little extracellular tissue in between the individual cells, or no matrix found therein. On the middle, you see the fibroblasts. So you find these mostly in cartilage and other connective tissues. And this is almost the opposite of the epithelial cells. There are actually less cells, but a lot more intracellular matrix. And this could be something as spongy as cartilage, or could be something as hard as bone, or something as fluid as we see under the blood cells through here. And we see two different types of blood cells here. You see erythrocytes, which are going to be without nuclei, which are going to carry the hemoglobin for oxygen transport. You'll find lymphocyte here, which is known as a white blood cell, more for protection and immune response. In this case, the matrix itself, otherwise known as the plasma, is somewhat fluid, and basically the blood cells, whether it be the erythrocytes or lymphocytes, are free floating. So again, closer look at the epithelial cells through here. So you see, which is known as the epithelial tissue. If you follow the mashed up together nuclei down on the bottom there, it gets really dark, and then it actually transitions to a different type of tissue underneath that. That's known as dermal tissue. The dermal tissue and the epithelial tissue actually will fit into each other almost like a jigsaw. This helps keep your skin somewhat elastic, but doesn't allow it to tear. So very simply, this is another protective organism that a lot of animals in particular have to basically protect themselves from the outside environment. We see the fibroblasts, again, we talked about before. So we find these in cartilage or in bone. So this can range anywhere from a nice spongy material that we find in cartilage, or sometimes in the tissues found in padding around different types of organs, say like around the heart, or say around the spleen, or even around the organs. This can include fat tissue as well, where oftentimes that matrix there, instead of being matrix, is actually going to be fat deposits found within the cells themselves. They look almost like diamond rings, where basically the entire portion of the animal cell is almost entirely engrossed in fat droplets or lipids, whereas the nucleus and the rest of the organelles are sandwiched out to the side, based up against the plasma membrane. In this particular section here, it looks like the fibroblasts are made into fibrocartilage, which is the kind of cartilage that we see in the intervertebral discs, or the elastic cartilage we find in the ear and also around the nose. And again, here we see the blood cells again. So the white blood cells tend to be fairly larger. This one is a lymphocyte. There are other types of white blood cells too. There are be more for particular types of processes or different types of invaders. We have specialized blood cells for parasites. We have different blood cells for the histamine response. So those of you that get reactions, allergic reactions, you can thank certain types of white blood cells for that particular reaction. We also have monocytes, which tend to be phagocytic. If you remember, those are the ones that actually will consume different types of bacteria once they're tagged by the lymphocyte that you see here, once it secretes any kind of antibodies. But you find these all throughout different types of animal systems. The blood cells here are human blood cells. They do tend to have somewhat of different shape in different types of eukaryotes, and also made different colors. If you look at insects in particular, they don't necessarily have hemoglobin. Bound to iron, they have different types of scarce metals that are used to bind different types of oxygen all throughout their particular vascular systems too. But again, this is a pretty good representation of what you normally see as far as specialized cells found in this type of tissue.